The overall goal of this protocol is to analyze the extracellular matrix protein composition, including the identification of glycosylation sites and compositional characterization of glycan forms in cardiovascular tissues.
This method can help answer key questions in the process of tissue fibrosis, such as how extracellular matrix glycosylation affects tissue fibrosis during cardiovascular disease. The main advantage of this technique is that unlike previous protocols, which analyze overall glycosylation changes, it allows for the characterization of glycosylation at particular sites of extracellular matrix proteins, providing a tool for more targeted follow-up mechanistic studies. Demonstrating the procedures will be Dr. Marika Fava and Ms. Ferheen Baig from our laboratory.
To begin this procedure, prepare all extraction buffers as outlined in the text protocol. Next, use a scalpel to dice 20 to 50 milligrams of tissue into 3 or 4 smaller pieces about 2 millimeters in size. Place the diced tissue into a 1.5 milliliter tube and add 500 microliters of ice-cold PBS. Wash the tissue 5 times using 500 microliters of PBS for each wash to minimize blood contamination.